Okay, nucleic acids, here we go. So you're currently looking at one of my normal PowerPoints that you see. I got a lot of words on there, and I've also got some diagrams. Right here, we've got the one on nucleic acids. The one you're looking at in this picture right here, if you can see my mouse pointer, is DNA, deoxyribonucleic acid. So you can see it's got these big blue backbones going around it, and on the inside you can see there are all these little shapes. That may not make any sense right now, but if you remember way back in September and October, we talked about what those were. And these are what they call nitrogenous bases. This is the sugar phosphate backbone of DNA, and we'll see the repeating patterns that make that up in a moment. So what a nucleic acid is, they're found in the nucleus. That's why they're called nucleic acids. They store the code for proteins, which boils down to all your genetic information that's in your body or that you'll pass on to your kids. They're made of nucleotides. If you remember, any of these big macromolecules are all polymers. And the monomers that make up these polymers, in this case, are called nucleotides. Moving on. The ones we're actually talking about today, the main nucleic acids you need to know about are DNA and RNA. DNA is deoxyribonucleic acid, and RNA is ribonucleic acid. We talked about that earlier on, but again, just to refresh your memory. So if you recall what the difference is, take a moment, pause this recording, I'll wait, and I want you to write down what you think the difference was. Okay, so pause this now and write down your answer. Okay, welcome back, and we'll see if you're right a little later on. So, one thing I want you to notice about these two molecules that you're seeing here, this one over here, which is DNA, and this one over here, which is RNA, is the number of strands they have. So you can see that DNA has two strands, and they're wound around each other. There's this one starting over here, and then there's one starting over here. They wind around each other. And if you were to untwist that, it would look more like this over here, into like a straight, almost ladder-like conformation. Over here, if you look at RNA, it's only one strand. It's one strand that loops around and does bond together with itself by these hydrogen bonds right here. But a lot of these, you can see over here, a lot of these nucleotides, that's what the little letters and shapes are. Each one of these represents a nucleotide. That's a C nucleotide, that's an A nucleotide, and so on. They're not paired up with anything right now. If you look at DNA, you can see they're always paired. So just remember that. Moving on. So if you look at an actual nucleotide, which is the monomer for all of these things, whether it's DNA or RNA, there are three major parts, and I color-coded them here so you can see them. The first one you're going to look at is a 5-carbon monosaccharide. That's right here. In this case, it's deoxyribose. That must mean this is a DNA nucleotide. If this were an RNA nucleotide, that would be ribose instead. But either way, you've got that 5-carbon monosaccharide in blue. Second thing is a phosphate group. You can see that right here. It's yellow. The phosphate group is part of the backbone. We always call it the sugar phosphate backbone. And if you look down here in this picture, you can imagine this being a piece of that big ladder that I showed you earlier. And you'll see this again, so don't worry too much. Notice the color coding stays constant. This is the sugar molecule right there, and up here is the phosphate right there. That leaves this little green guy right here, which you looked at in the previous slide. If you want to look back at that, you can pause this at any time and go back. This little green guy right here is a molecule called thymine, and that's a nitrogenous base. Number 3 right here. They're called nitrogenous because they have nitrogen in them. And if you look, you can see that little N, guess what that stands for? You guessed it, nitrogen. They have a lot of those in there. So those are the three parts. They're color-coded. 5-carbon sugar, phosphate group, nitrogenous base. That's a nucleotide. These are two of the 5-carbon sugars that we were looking at before. I want you to pause this, but before you do that, you're going to tell me which one is ribose and which one is deoxyribose. So I want you to pause it and write down in your notes which one you think is deoxyribose and tell me which one it is. Go. Alright, welcome back. So hopefully you figured this out. The major difference you should have been looking at is, notice these numbered carbons? I wrote that up there. See ya? 1, 2, 3, 4, and 5. They're numbered for a reason. Look at the number 2 carbon on both A and B. On the number 2 carbon you see over here a hydroxyl group on the B molecule. On the A molecule you don't see that oxygen, you only see a single hydrogen. So that must mean this is missing an oxygen, or deoxyribose. So this one's deoxyribose, That one is ribose. Hope you got it right. These are the bases. So these are actually what make the code for proteins. Each one of these bases has a letter that's assigned to it based on what it's called. So thymine we call T, it's the first letter of it. Cytosine is C, adenine is A, guanine is G. So you can see these all have a little bit different shape. They're slightly different, even though C and T look kind of similar, but you can see by the things that are bonded to them, they're not quite the same. Same with adenine and guanine, not quite the same, but they do look similar. Over here in its own box is uracil. Uracil you only find in RNA, and it takes the place of T. So there's no thymine in RNA. But since we're mainly focused on DNA, we'll stick to these over here in this box. The double-ringed ones that you see right here are called purines, and the single ones are called pyrimidines. So even though the one that looks the biggest has the shortest name, and the one that's the smallest has the longest name, maybe you'll be able to remember it that way. So A, C, T, and G, adenine, cytosine, thymine, guanine. Remember those. There's a certain order that they pair up in. Remember, if you looked all the way back in the beginning, let's go back real quick, you can see they pair a certain way. Notice A and T and C and G are always together. You're not going to fit it any other way. If you notice, here's one of the things we'll talk about in a second, but the A molecule, or the A nucleotide, looks like it's a little longer than the T is. Just like the G is a little longer than the C. If you paired an A and a G together, that would make it too wide and this molecule wouldn't be straight anymore. If you paired a C and a T together, then it would be too short, so you'd have an uneven looking molecule. Anyway, going back to where I was, right here. If you remember what we learned about Erwin Chargaff, you'll remember that he came up with rules of which nucleotides would pair with each other. And there's a reason they pair the certain ways that they do. Purines will always pair with pyrimidines. So you have one big molecule with a, oops, my fault, let me go back. You always have one big molecule with one short molecule. Again, one big one, one little one. So A always goes with T every time. Adenine and thymine go together. And you can see these dotted lines right here represent hydrogen bonds. And if you remember what a hydrogen bond is, it's a weak attraction between a negative and a positive part of a molecule. So you can see right here, this O is slightly negative, this H is slightly positive. So they will attract each other. Same here. This H right here and this nitrogen over here will attract each other. And there's only two places where these bonds can form, so adenine and thymine will always be together. Then if you look at C and G, they're always going to be together because they can form three hydrogen bonds between this O and this H and this N, this H and this O. So because this opportunity is here, all three of those bonds will form. So you will always find C and G together. You will never find G with T. If we were looking at RNA, where you normally see A and T bonding together, U would take the place of thymine right here. Okay, here's Chargaff's results. The reason why these are important is because if you look, no matter what the organism is, here's the source that he used, Streptococcus, which is a bacteria, yeast, which is a fungus, herring, a fish, and a human. If you look at the proportion of adenine and thymine, you'll see that 29.8, 31.6 are pretty close. These numbers are always very close to each other between A and T. If you look over here at C and G, they are also much closer to each other than they are to either A or T. Hopefully that makes sense. But the point of this was that when Chargaff looked at his results, he decided that G and C were probably the ones that paired with each other, as well as adenine and thymine pairing with each other. And he actually met with Watson and Crick when they were figuring out the structure of DNA and shared these findings with him. He didn't really like them very much, but apparently they used his work when they did it. Here's the DNA molecule. Again, if you look over here, this is the double helix right here. If you were to untwist it, you can see each individual nucleotide with its three component parts. If you remember what those three parts are, they're right here. There's the sugar, the 5-carbon sugar, which is blue, the phosphate group, which is in yellow, and the nitrogenous base right here, which can be multiple colors, but it always has a letter. You can see each one of those parts right here. Here's a T nucleotide zoomed in, there's a T nucleotide right there. There's its sugar, there's its phosphate group, there's its base. Pretty complicated, but I think you can do it. So here's what it looks like. If I zoom in on this molecule, I get something that looks like this. So this is a string of nucleotides right here, not paired up with anything else right now. But if you look at it, you can see where they would fall in place. So here's an individual nucleotide. If I zoom in on that, you get something that looks like what I showed you before. The phosphate group is actually usually represented with a circle. Sugars are represented with pentagons, at least if it's a 5-carbon sugar it is. And over here is a nitrogenous base. So if you look at this picture, you can see, zooming in from big all the way down to small, what these things look like. This is one nucleotide. Okay, I told you before that the carbons were numbered on that sugar molecule. So there's a reason why they're numbered. And first of all, I want you to look at this one. This is the number 5 carbon up here. At the number 5 carbon, you will always find the phosphate group attached. That might be worth writing down. And over here at the number 3 carbon, you will find another phosphate attached. So this will go on and complete the chain, and you'll get the next phosphate, and then following that, the next sugar. So if you remember, here's the phosphate group right there. There's the sugar. The phosphate group is attached to the 5 end up here, and then down here you're going to have another phosphate group, a little yellow rectangle. Don't believe me? Go back and look. There it is. You can see they just alternate back and forth. And where they're attached, we call this the 5' carbon, and this is the 3' carbon. So, going back to nucleotides, the only thing that makes each nucleotide different, we just talked about the sugar and phosphate backbone that they have. That just alternates over and over again, sugar, phosphate, sugar, phosphate. The nucleotides themselves are only different because of the bases that they have, whether that's A, C, T, or G. Here's the 4 bases again. They can either be purines, these two, or they can be pyrimidines based on that carbon ring structure they have. So this one has a single ring, the purines have a double ring, like that. Purines only pair with pyrimidines. Here's the base pairings again. They call this complementary, and I think you remember we used the term complementary before when we talked about photosynthesis and cellular respiration. What it means is that they are counterparts to each other. So you can see again, purine pairing with a pyrimidine. It's governed by these 2 hydrogen bonds, so A will always be with T, and C with G. There's 3 bonds between C and G, 2 bonds between A and T. That's important. You should remember it. If you remember when we just talked about the different carbon numberings that are involved with that ribose sugar, here it is again. Remember I said there's a phosphate group at the 5' end, and there's a phosphate group at the 3' end. Same with this one. That's the 5, that's the 3. 5, 3. You say it now. Nice job. Anyway, so this little apostrophe you see here, we say is prime. So this is the 5' end. So the way this works, if you look at this is what they call the 5' end of an entire DNA molecule. If this was the total end of the molecule, it stopped right here, they call it the 5' end. The other end, if you look, it's going to end at this hydroxyl group, and that's going to be at a 3' end. The other side is going to be the opposite. So the 5' end will be on this side, and the 3' will be on that side. So they call that anti-parallel orientation, because they are parallel to each other. There's one on this side and there's one on this side. They're parallel. But the anti refers to the fact that one of them is upside down when you compare it to the other one. And that's why these nucleotides are written in upside down. That just makes the point. And this is the double helix. This is the structure of DNA, and this is very similar to the model that Watson and Crick made with wire and cardboard. Now, this is all color-coded, but you can see right here, it kind of looks like a ladder that you twisted, and you end up with this shape. So in this case, the ropes that make up the ladder would represent the sugar-phosphate backbone, and these rungs on the ladder will represent the nucleotide bases. And that's it. So hopefully you took decent notes. You should have been writing all this stuff down. If you need to replay it, rewind it to anywhere you need to, and play it again. I expect to see your notes tomorrow when you bring them into class. See you then.